This is an overview video for the Cumulative Effects Modeling for Prioritizing Recovery Action, or CEMPRA toolbox. The CEMPRA toolbox was developed by a large number of collaborators to support cumulative effect modeling for aquatic species. The goal of this tutorial video is to understand the background and purpose of the CEMPRA tool, become familiar with the R-Shiny and R-Package version of the toolbox, and to understand how applied the CEMPRA toolbox to different contexts. So what is the CEMPRA toolbox? The CEMPRA toolbox is designed to be a flexible production-grade R-Package and R-Shiny application to support cumulative effect modeling geared towards aquatic species for planning recovery efforts. The generalized workflow is as follows. First, we start with a series of locations, or various spatial units of any scale. Next, we summarize stressor variables within each of these locations. Stressor variables can be assembled from GIS layers or from field studies. We try to represent these creatively to get the best representation of each stressor within each location. Finally, we define stressor response relationships and then compile these to generate cumulative effect scores. Then we can run the models again with either different stressors or different stressor response relationships to represent alternative future scenarios. The idea is to do this whole process in an iterative workshop type setting. Some elements of this workflow will be familiar from other cumulative effect assessments. The CEMPRA toolbox contains a simple cumulative effect roll-up component dubbed the Joe model throughout the documentation and the remainder of this video, and also a more integrative life cycle model. These two elements both have different endpoints and different use cases. The core components of the CEMPRA toolbox include the following. First, we have to think about a study system. So this will be a focal species or aquatic ecosystem of interest. Next, we think about a series of stressor response functions linked to the study system. So these could be things like thinking about turbidity in relationship to survivorship or temperature or habitat. Next, we think about a series of stressors that we can quantify or approximate that are linked to the stressor response functions. Finally, we'll have a series of locations. These are different spatial units that we can represent the stressor and study system at. These can be of any scale and may include subbasins, watershed, or even mesohabitat units within a river. Finally, we have a series of different assessment endpoints that we can evaluate. Namely, this might just include a cumulative effect roll-up or estimates of capacity and productivity from our life cycle model. Finally, we combine all these elements into different scenarios. So these might be using the tool to try to represent different types of restoration action or potentially future alternative development scenarios or climate change. The whole CEMPRA toolbox is based around stressor response functions. Stressor response functions are the foundation of this toolboxing framework. Stressor response functions are also referred to as dose response curves, but mainly they include anything that we have a raw stressor variable on the x-axis and a biological response on the y-axis. The example from Jensen below shows the percent fines in habitat on the x-axis and then the biological response for egg to fry survival of very specific salmon on the y-axis. So here we have our standard template of a stressor response relationship with the raw stressor values on the x-axis and the biological response on the y-axis. Numerous examples of stressor response relationships can be found in the literature. These may include simple threshold values, a functional curve or response, or minimum and maximum bookends, potentially with some curve on the relationship. When we're thinking about stressor response relationships, we have to think carefully about what is the actual biological response. In some cases, it may be very well defined, like something like egg to fry survivorship from fines. However, in other cases, the biological response may be more generalizable or more complex given multiple pathways. It may be more appropriate to find a way to standardize it. We'll come back to this later when we think about the differences between the life cycle model and the Joe model for cumulative effect roll-ups. In the example below, we can see a series of locations color-coded according to their stressor response relationships and stressor magnitude scores for two different stressors. So there's three main data inputs in the CEMPRA toolbox. One is the stressor magnitude file, two is the stressor response file, and three is the location. We'll come back to the specifics of these later, but what's important to note is that the format of these inputs allows for stochasticity in both the stressor magnitude and the stressor response relationships. So within the CEMPRA toolbox, we have three different types of assessment endpoints that we can look at. All of these are based on the stressor response functions. The first assessment endpoint is just thinking about individual stressors, so just mapping them out with univariate response plots. This can be useful to look at the distribution of stressors across the landscape or study area. The next assessment endpoint is the Joe model, which summarizes these stressors using a simple cumulative effect roll-up score. Finally, we can try to incorporate the stressor magnitude data and the stressor response relationships into a life cycle model. So the individual stressor plots can be used just to look at the distribution of stressors across the landscape. The Joe model, we'll go into a little bit more detail behind this later, but it's just a very simple cumulative effect roll-up score. So it's just taking the product of the response relationships for each stressor. Finally, there's the life cycle model, which integrates the stressor magnitude and the stressor response relationships into a stage-structured matrix model to look at components of capacity and productivity for different stressors or scenarios. We'll cover the life cycle model more in another video. So first, we'll just cover the Joe model, which is a simple cumulative effect summary. Again, thinking about the Joe model in context to the life cycle model, the Joe model is a bit more simple and easy to get set up with, but it can be extremely useful in workshop settings to quickly run through a large number of stressors without getting too hung up on all the technical details. So the Joe model itself just takes the product of the response scores from a series of stressor response relationships. In the example below, we can think of the black dot and the yellow dot representing two different locations or potentially two different scenarios for the same location. The Joe model just takes the product of the different stressor response relationships. So it strongly penalizes any individual location that has an extremely low score for one stressor. The Joe model characterizes the cumulative effect score across all stressors is the cumulative system capacity. This wording is intended to be generic, just to kind of put different stressor response relationships together in a workshop setting and just to try to move forward with a large number of locations and a large number of stressors without getting too hung up on the technical details behind any one stressor or stressor response relationship. Within the application itself, there's a web map interface that lets you flip through different stressors to display different heat maps for them, and this lets workshop users kind of understand like the distribution of stressors and scores across the landscape or study area of interest. One thing that's really important and really powerful about the Joe model is that the structure of the stressor magnitude file and the structure of the stressor response relationship file has standard deviation, allows for stochasticity in both the dose and the response curve. So the stressor magnitude file characterizes the stressor values for each location. We can see that it's actually represented in a long format rather than a wide format. So for this Huck number 170, there will be a stressor called August stream flows with a mean value of 100, and in this case, 0 standard deviation, but then a lower and upper limit for resampling. In the stressor response relationship, we also have a standard deviation and lower and upper limit. When we run the Joe model, we actually generate a distribution of values for each stressor in each location. So here in this example, we can see a number of different locations with individual plots, and then their cumulative system capacity score, just the mean value shown as the red line. And then these are just histograms here showing the distribution of values from a resampling exercise. So this can be valuable to, depending on the level of stochasticity in either the stressor response or the stressor magnitude values, to really kind of understand different locations or units according to not only their mean value, but also the distribution and uncertainty. We can also run batch processing with the Joe model to quickly kind of compare scores across different scenarios where the locations and stressor response relationships may stay the same, but different stressor magnitude different stressor magnitude data inputs may be used to represent different scenarios. We can also compare the distribution of scores across individual location units or basins there. The life cycle modeling framework is a bit more involved. I might make a whole separate video on it later, but the idea behind it is that if certain stressor response relationships are very well defined and they're linked to components of the life cycle, then it might be valuable to try to represent each of the stressor response relationships into a life cycle modeling simulation, and to see if that may produce a different result than just the cumulative effect Joe model rollup. So the life cycle model within the CEMPRA toolbox, it was developed in part by Kyle Wilson based on a series of other studies from Martin Koops and Adam Vanderlee, but it's at its core, it's a stage-structured population model. So it looks at transitions between different stages. And the idea is to introduce stressor response relationships into the model, link them to different vital rates, and then to kind of compare different population level metrics across scenarios, such as carrying capacity, viability, and productivity. So with the life cycle modeling component, we still have the stressor magnitude Excel workbook and the stressor response Excel workbook, but we also introduce a new species profile data set. We'll go over this in a bit more. We run the life cycle model, and then we look at relative changes to productivity or capacity across different scenarios. The life cycle modeling input file is designed to be a standardized CSV file with a series of parameters, names, and values. The name field is standardized, but you can add any number of life stages according to one of these template files, and then introduce that to run the life cycle model. Each life cycle profile is meant to represent a single population. So here's a generalized life cycle profile for Athabasca rainbow trout. This is the input CSV file, and then under the hood, it's running a stage-structured population model. So in the CEMPRA toolbox, the life cycle profile builds a stage-structured population model, and then the stressor response relationships are linked to various components of either survival, fecundity, or capacity as the life cycle model runs. So for instance, if we have a stressor such as temperature, and that influences fry survivorship, you might start with a default fry survivorship of, say, 20% or so, and then depending on a given location's stressor magnitude scores, the default survivorship may be decreased by some percentage. Likewise, the carrying capacity for a location for a specific life stage may be decreased or increased by a stressor, and other stressor response relationships may be linked to the fecundity as well. The population model runs and it generates a simulation or a time series of the population over time. And then from the different time series, when you rerun the model under different scenarios, we can then start to think about population level metrics, such as like relative changes to the equilibrium carrying capacity of results. Equilibrium carrying capacity across the different scenarios. So in this scenario for Athabasca rainbow trout, in red here, we see a baseline scenario, and then scenario A and scenario B could change a series of stressors to represent restoration actions. And then we use the life cycle model to look at the relative change above baseline there. And so just as a bit of a caveat to the life cycle modeling component of the CEMPRA toolbox, it's designed to be pretty generalizable and simple. There's a lot of other alternative life cycle or population modeling tools out there, but the idea behind this is to kind of use it in a more generalizable and standardized framework, which means that there's some strength to be able to apply it quickly to a new case study area or problem, but some specific customizations, which we see commonly in various population modeling exercises, might not be able to be represented here. So here I've just kind of listed some of the pros and cons here and sort of where the life cycle modeling framework in the CEMPRA toolbox lands. So next we'll look at the CEMPRA R Shiny application and just do a bit of a walkthrough here.